Good day, everyone. I'm Atayelu Marquiza from the Group 2, and I'm going to report or discuss the infection caused by anaerobic bacteria. So, the report covered introduction, definition, and classification of anaerobes, anaerobic bacteria as commensal flora, diseases caused by anaerobic bacteria and site of infection, virulence factor, pathogenesis, incident of infection, laboratory diagnosis, antibiotic susceptibility test, and the antimicrobial activity. Introduction. So anaerobes are indigenous flora of skin and mucous membranes, cause infections involving every organ and anatomic region of the body. So here are some definitions. So anaerobes, bacteria that requires anaerobic conditions to initiate and sustain growth. Strict anaerobes, unable to grow if more than 0.5 oxygen. Moderate anaerobes, capable of growing between 2 to 8% oxygen. Micro Microphilic bacteria grows poorly in air but better in anaerobic conditions. And lastly is the facultative bacteria, grows both in presence and absence of air. So here are some classification of anaerobes. So it has two gram, the positive and the negative. So in positive, in bacilli, it has two also classification. It's sporing and non-sporing. In cocci have Peptococcus, Peptococcus, and Peptostreptococcus and Feingoldegg. So in negative, the bacilli is the Bacteroides, Prevotella, Porphyromonas, Fusobacterium, Leptotrachea, and in cocci is Veillonella and Megasphaeria. And also the so so the anaerobic bacteria is commensal flora. It has a oral cavity and upper respiratory passages, stomach and small intestine that has a lactobacilli, large intestine and terminal ileum has Bacteroides fragilis group. The GUT or vagina and cervix have pigmented Prevotella, urethra in both male and female that has Propionobacterium, and also the skin. Next is the disease caused by anaerobic bacteria. We have the mouth, head, and neck. It has a sinusitis, otitis media, and many more. Also the thoracic cavity that has empyema, lung abscess, also the abdominal cavity that has peritonitis and intra-abdominal. Pelvic cavity or vagina has PID and pelvic abscess, bacteria, vaginosis. Also the skin and soft tissue infection has diabetic foot ulcer and cutaneous abscess. And lastly, the central nervous system, brain abscess, subdural empyema, and epidural abscess. So this is we can see the sites of anaerobic infection. So makita na to sa itong mga lawas kung asa dapit ang anaerobic infection like dental infection, that ang kanang mga infection from bite, also mga foot ulcers, so naa-jud siya. So also the brain abscess and yan. So morning table sa virulence factors associated with pathogenic anaerobes. So incident of anaerobes in various infections. So these are the type of infection: lung abscess, necrotizing pneumonia. It has the incident percent of 62 to 93%. Bacteremia has 6 to 10%. Brain abscess has 60 to 89%. Chronic sinusitis has 52%. Thoracic empyema has 76%. Intra-abdominal or pelvic abscess has 60 to 100%. Perical abscess has 75%. Gas gangrene has 85 to 95%. Post appendectomy has a 40%. So kanang mga red numbers is mao nang mga incident nga mga infection nga daghan kay mga incidents. So next is the laboratory diagnosis. So specimen collection and transport specimens suitable for anaerobic culture that has a material aspirated from abscesses, the decubitus ulcer, sulfur granules from discharging fistula, tissues obtained at biopsy or autopsy, fluid from normally sterilized site, pulmonary specimens, the gastrointestinal specimens, genitourinary specimens, and also the blood, bone marrow, and CSF. We have also the specimens unsuitable for anaerobic culture. So we have bronchial, wash or brush, cough, sputum, disease, gastric or small bowel contents, elastomy or colostomy, drainage, nasopharyngeal swab, throat swab, rectal swab, urethral swab, vaginal or cervical swab, secretions obtained by nosotracheal or orotracheal suction, voided or catheterized urine. In aspiration is ideal, avoid swabs. So collection by needle aspiration is preferable than swab culture because it has better survival of pathogen, great quantity of specimen, and also less contamination with extraneous organisms are often achieved. So we have a transport system as transport media, PRS, or the pre-reduced anaerobically sterilized, syringe and needle sealed with a sterile rubber stopper, CO2 vials, Bactec anaerobic bottle for blood culture, Vacutainer anaerobic transport, and the hangit tube. So this is the starch transports media. So we have a common anaerobic media. We have non-selective, the liquid media, and the selective. So we're going back to the non-selective. In non-selective, we have anaerobic blood agar. The non-selective medium for isolation of anaerobes and facultative anaerobes contains 5% ship blood, hemine, L-lysine, and vitamin K1. Next is the supplemented brain heart infusion agar, enriched with addition of yeast extract, vitamin K1, hemine solution, L-citrate. Next is the liquid media. So we have the Robertson cooked meat broth, pepton yeast extract broth, thiolate broth. So in Robertson cooked meat broth, the ground beef yeast extract, trypticase KH2PO4, lysine, and resazurine solution distilled and distilled water. And the pepton yeast is the non-selective for cultivation of anaerobic bacteria for gas liquid chromatography. And the last one is the for cultivation of anaerobes as well as the facultative anaerobes and aerobes. So next is the selective. Bacteroids bile esculent agar is the or BBE, selective and differentiate for bacteroids fragilis group. Number 2 is the lake kanamycin, vancomycin blood agar or LKB, selective for isolation of Prevotella and bacteroids group. The third one is the cycloserine, cefoxitin for fructus agar or CCFA. It is a selective for Clostridium difficile. Right. So number 4 is anaerobic phenylethyl alcohol agar or the PEA, selective for inhibition of enteric gram-negative rods and swarming by some Clostridia. Next is the methods of anaerobiosis. So anaerobic system for cultivation is conventional anaerobic jar with evacuation and replacement of gases with hydrogen, nitrogen, and carbon dioxide. Gas pack anaerobic system, the anoxomat, the PRAS, the PRAS or pre-reduced anaerobically sterilized media, and anaerobic cabinet and glove box. So anaerobic incubation is 85% in nitrogen, 10% of hydrogen, and 5% of carbon dioxide. So this is the anaerobic jar techniques. So jar are used primarily with primary plate, media, or subculture plates. Oxide jar has a metal lid, valve, and a pressure. It can be used either as the evacuation replacement jar or it can be used with a disposable gas generator, the gas pack. Next is the anoxomat. So mauna siya. And also the gas pack. That's it. And we have the prime and the anaerobic glove box. So kanang anaerobic glove box na nasa to ang lab kanina. And also the anaerobic disposable plastic bags, and the roll strike system. So there are 5 methods for diagnosis of anaerobic infection. It has direct examination of specimens and staining, culture, metabolic product, detection by gas liquid chromatography, molecular methods like PCR, and also the rapid system. So first, let's discuss the direct examination of clinical materials. So a food, odor, purulent, appearance of fluid, specimens, and presence of necrotic tissue and gas of or sulfur granules are valuable for suspicion of anaerobes. Background and cellular characteristic of smear, a acridine orange stains are useful for detecting bacteria in blood culture, CSF, pleural fluid, joints, fluid, and exudates. Next is the direct microscopic examination or the gram stain. Polymicrobial infection, characteristic of anaerobic bacteria and multiple distinct morphotypes of gram-negative and gram-positive bacteria suggestive of anaerobic infection. Bacteria seen in smears but no growth when cultured aerobically. So, ano ang bacteria? So, kani siya ang bacteroids, the Poropheromonas prevotala, faintly staining gram-negative Coccobacilli. Next one is the Fusobacterium nucleatum, thin gram-negative bacteria with tapered or pointed ends. Next one is the Clostridium perfringens, large boxcar-shaped gram-positive bacilli with blunt ends. And also the Veillonella, tiny gram-negative cocci with gram variability. Next one is the actinomyces, branching filamentous bacilli with bedded gram-positivity. And the bacterial vaginosis. So makita dira si gram variable and si Coccobacilli. Next is the culture. Colonial appearance of common anaerobes on culture media. So first, this is a colony of bacteroids, fragilis, on anaerobic blood agar. It is a non-hemolytic, gray colony with concentric whorls inside. And also, next is the bacteroids, fragilis on bacteroids pilesculin agar or the BBE. It has also the Poropheromonas SPP on anaerobic blood agar. Tan to buff colonies, brown-black pigment, brick red, fluorescence, or UV. So maklaro man ang no? And next one is the Prevotella. Brown to black colonies on BA, brick red, fluorescence, long wave UV, produce indule and the ferment glucose. Next is the actinomyces. Colonies of actinomyces Israeli are 0.5 or 2 millimeters in diameter, white or gray to white, smooth, entire, or lobulated, resembling like a molar tooth. Next is the growth of yellow colonies and Clostridium difficile on Cycloserine cephositing fructus agar, or in short, the CCFA with horse stable odor. Next is the Clostridium perfringens, double zone of hemolysis on blood agar, smaller zone of complete hemolysis due to the theta toxin, outer zone of partial hemolysis due to alpha toxin, with a pacification of egg yolk agar due to the phospholipase. Next is the Clostridium septicum, gray to translucent, markedly irregular swarming. So over the surface of blood agar will underlying be hemolysis, beta hemolysis. And next is the gas liquid chromatography. It's used to detect anaerobes in exudate and fluid body. A major amount of butyric acid on a specimen that contains only thin, pointed gram-negative rods would suggest Fusobacterium sp. So a major peak of succinate and the presence of only gram-negative rods would suggest bacteroids. A major propionate peak in a positive blood culture contains pleomorphic, non-spore-forming gram-positive rods would be most consistent with Propionibacterium sp. So, and lastly is the direct GLC. Provide only presumptive clues and should be interrupted occasionally in polymicrobial. And the D is PCR. PCR amplification procedure appear promising but are not well commercialized. Anaerobes identified by colony PCR and sequencing of the 16S or RNA gene using universal primarily. And the molecular diagnosis for Clostridium, detection of phospholipase gene PCR detection of alpha, beta, epsilon, and iota toxin gene by multiplex PCR. Next is the rapid methods for diagnosis of anaerobes. Two rapid systems are available for quick diagnosis of anaerobes. So, naidyo ka buok. So, this is the rapid ANA by Innovate Diagnostic System and by analytical product. This both system rely on performed enzymes and only 4 hours of anaerobic incubation is required. This advantage is costly and variable response. So, mao ni ang presumptive identification of anaerobic gram-negative bacilli. Next is the anaerobic gram-positive cocci presumptive identification. Gram-positive, gram-variable, or gram-negative cocci or cocci bacilli, and also the Peptostreptococcus and anaerobious growth inhibition by sodium polyanethyl sulfonate. And lastly is the definitive species identification of anaerobic bacteria. Biochemical reaction in pre-reduced anaerobically sterilized or the PRAS liquid media. And the fermentation and product analysis and or cell wall fatty acid profiling by gas liquid chromatography or the GLS. And also lastly the 16S RNA gene sequencing. So maragtaas ang report nta namin mo, and thank you. If you have any questions, don't hesitate to ask for